recording the lecture from now. Even though there's no video for some reason on my side, I'll start with the sharing of the screen. Sharing of the screen. There. Isn't that weird? Okay. Today I've got lecture 9 up.
Let's be out. I question it a lot.
Lecture 9 is environmental factors. And I've got an assignment 4 for you guys. The assignment 4 essentially has 2 parts. One part where it kind of reviews the idea of a growth curve. And the second idea there is how to do the lab this week. In the lab this week, you've got concentrations, and we have to kind of count for the concentration dilution factors. So that's what the assignment is today. If you're online and you're looking for the assignment, it's assignment 4. I've posted it online. There should be a link for it. Also, reminder, today's test. So after class, go home or wherever you have a laptop, access to a laptop, do your test. Be sure to get it done today. Okay. I've set the deadline for the test to be like after midnight sometime. So, okay, so if you're working this evening, you have till whenever to finish it off. Okay. Okay, assignment 4, I'll leave for the end of class. Two things here. The title slide here has a theoretical growth curve of what a growth curve for some microbes looks like. And as you can see, there's a little tiny lag phase, an exponential phase, stationary phase, and what's referred to as a depth phase. That's a great theoretical sort of model. But if you actually did an experiment where you're growing bacteria or yeast or some sort of exponentially growing microbe, it doesn't look like that at all. When you actually grow it, or oh, is this recording? I should check. I did? Yes, okay, it is recording, good. Okay, so I distracted my own thoughts. What was I saying? I was saying these numbers are real, but they don't look like the theoretical growth curve so much. So the assignment that I'm giving out is like a assignment where I'm asking you to show me some real numbers, okay? Okay. Let's carry on. Okay, so Aaron and who else was online? Participants, there's 3 participants. There's the chat. Where's my list of participants? There. Here's a brief overview of the content for the test. So the last 4 lectures, lecture 5, lecture 6, lecture 7, and 8, I covered in lecture 5, counting microbes, things like solid media, referential media, selective, differential media, different ways of making isolation counts, direct count methods. That was all in that lecture. And then lecture 6 was germ theory. I talked about 2 individuals mainly Robert Koch and Louis Pasteur, and there are two experiments. The boiled beef experiment, which I asked you to kind of remember, and the Robert Koch's 4 postulates on how to Koch's postulates, how to show that an animal actually is getting poisoned by a microbe, and how to recover that microbe and how to kind of pass it to another animal to prove that that's the microbe that caused the disease. And we talked about anthrax, tuberculosis, cholera being a couple of examples, and rabies being a couple of examples of their work. So that was germ theory. Also the theory that germs cause diseases or microbes cause diseases. That's the essential definition for germ theory. Then there's food pathogens and sterilization. I introduced sterilization with the story of Maple Leaf Foods and the Listeria outbreak, as well as the Walkerton water treatment plant incident where a number of 6 people died from drinking water contaminated with Escherichia coli. The test doesn't focus too much on Maple Leaf Foods or Walkerton or even botulism so much, but it does talk about or ask you to try and recall the microbes involved in diseases, the food diseases. So remember, I gave you a quiz 3 or assignment 3 where I ask you to identify the food microbe that and the disease it goes with. That's going to be on the test. Okay. Last thing, food sterilization and disinfection. There's a number of treatments to disinfect food. Okay, and so that's a brief overview of what we're covering for the test. Any questions? Okay, so I didn't make any review questions for my list of people: Natalie, Jen, Adam, Danielle, Tobechku, Paul, Lindsay. There's no review questions, but there are questions. So, and the questions aren't too hard, I hope. Okay, so here's a lecture overview. The first part of the lecture is I'm talking about cell growth. Then I'll talk about other environmental factors that influence the growth of microbes, including temperature, and then oxygen, pH, and then finally a little bit about water activity. Water activity, I have to apologize in advance. This is a new topic for me. I've never studied water activity in another course, so what I've learned about water activity, I've actually taught. So you probably have taken it in other courses, right? Right, so good. So you guys have probably, yeah, that's what I'm hoping. Over the course of me teaching the course a couple of times, students teach me about water activity. Okay, so that's environmental factors for growth. That's what we're talking about today. So your standard growth curve has this hill shape, and the hill shape at the beginning, there's a little bit of a lag when the bacteria have been trying to accommodate their growth method for the media that they're in. And then there's this phase, it exponentially grows. And you can actually tell that it's exponential because what we do is we use log paper graph or logarithmic graphs. So we're not measuring in just base 10 numbers, like not 110 to 1,000. We're going by base numbers. So either base 2 or base 10, just to show that there is a linear phase of the growth that is actually kind of exponentially growing. And then once it finishes the exponential growth, the microbes kind of environmentally adapt to whatever media they're in, and we refer to that as a stationary phase. And so they're able to maintain healthy colonies for a while. And then at some point in time, a lot of the material within the media has been degraded. And so the microbes start dying off. And this is referred to as a death phase or a killing phase. Yeah.
Are you talking about the finite media, like,
Yep, great.
or is it that something very big that the bacteria can keep on growing and growing?
For this kind of experiment,
Yeah.
it's a container and you have a fairly defined inoculum. It's usually a 1 in 1,000th dilution or something like that. When I say 1 in 1,000th, I'm thinking, so if the optical density, meaning absorbance using a spec, is 100% at 1, then you use a 0.001 inoculum. So if you have 1,000 liters, you would use a 0.001 inoculum, inoculate that, but it would be like the appropriate volume as well. Okay. Because if you have 1,000 liters, the lag phase is really long before it, if you only have like a very, very small volume. Okay, so in bacteria and yeast, the growth of the organism into 2 cells is like a binary fission. And in bacteria, the daughter cells are identical to the original cell. So you can't really tell which is which. But you have this occurrence where 1 becomes 2, 2 becomes 4, 4 becomes 8, and it's doubling at a certain rate of time. And that's the exponential phase, is the doubling time. So for a period of time, they don't double very quickly. That's the lag phase. And then there's the doubling time. And then when the population doesn't double so much anymore, that's referred to as a stationary phase. And then there's a decline. It's referred to as transition phase or death phase. Okay. I have here, is it death or just stasis for some select members? This is a question of active research. People actively look to try and figure out, is this cell really dead? Even like it might be static, it might look dead, might not have any metabolic activity, but if you take some of these dead cells into fresh media, boom, they start growing again. So it's a question for some researchers to kind of figure out, is it really dead or is it not quite dead yet? So the measurement of exponential bacterial growth curve is a training for usually what people do when they go into a research lab. Prof will say, okay, make me a growth curve of this bacteria. And they give a person, say, Escherichia coli, Yersinia pestis, or whatever the bacteria is. And the person has to watch the growth over a day or a week or a month, depending on the microbe. So some microbes, doubling time is in the terms of 10 to 20 minutes. And if a doubling time is like 10 to 20 minutes, then you're done your experiment within a couple of hours. If the doubling time is 12 to 24 hours, then your experiment is over weeks later, because it takes a long time for it to double. Okay, so, and we count the bacteria by direct counts. And by direct counts, this is like a microscope. So you'd have a microscope slide, put some bacteria on it, And there might be a grid, and you can actually count how many bacteria in the grid, and that grid relates to the volume of what you put onto the slide. And then what you do is you would calculate backwards, figuring out how much volume you put down from the original culture and how many bacteria that original volume represents. So that's a microscope way. Then there's also something called flow cytometry. This is a cool kind of machine. What it does is it counts particles, and it can count fluorescent particles, or it can count microbial particles. And what you do is you'd put in cells into a dilution, and this pipe would go through the instrument, and the instrument would detect the number of cells through using some sort of light-based measurement. So it would count all of the particles, and you get a number of particles in your solution. So you could actually figure out how many bacteria you have in your original. Another way is by how heavy it is. Remember in lab, you had the kombucha. You can actually figure out how much inoculum there was, how much kombucha there was by how heavy the pellicle is. So that's a measurement. That's referred to as a biomass calculation. Then you've got colony counting, which is a lab coming up for you tomorrow. I'm going to have you plate out some bacteria in 2 different ways, and you'll come up with a colony count for me in a week's time, telling me how many bacteria were in the original bottle. Then there's indirect ways as well. One is turbidity. Turbidity is based on how cloudy your liquid is. And what you're doing is you're shining light, you're taking the absorbance reading, and based on how turbid it is, it gives you this amount of bacteria. Okay, so that's the idea of growth in media. Then there's this idea of exponential doubling. So there's different base systems. There's the 2, you could have a base 2 numbers, base 3 numbers, base 4, base 8, just whatever number you want. That could be your base system. I was reading someplace a little while ago that, I don't know, was it Assyrians or Phoenicians? This culture from 4,000 or 5,000 years ago, they didn't have a base 10 numbering system. They did all of their calculations on a base 6. So all of their tiles, they'd have these clay tiles with all these numeric calculations, actually base 6 numbers. So, wow, it's kind of different. Yeah, very different. Your number system would look totally different than what it is now. There's also a base. So at the bottom of this slide, I've got a base 2. You see there's the example 2 to the minus 2, 2 to the minus 1, 2 to the 0. So if you think 2 to the minus 2 is 1 over 2 squared, because the minus means that it's the bottom of the denominator. So 1 over 2 squared, or 1 over 2 is like 1 over 4, 1 over 2, and then 2 to the 0 is just 1. 2 to the base 1 is 2. 2 to the base 2 is 2 times 2 before 2 to the base, or 2 to the exponent 3, 2 times 2 times 2. So it's just how many times you multiply it by. And I have the fractions right there: 1/4, 1/2, 1, 2, 4, 8, 16, and it keeps going double every time you go up. If you do it by 3s, it's 1 over 3 squared, 1/9, 1/3, 1, and so on. For 10s, base 10s, 10 to the minus 2 is 1 over 10 squared, or 1 over 100, or 1 over 10, 1, 10, 100, 1,000, as you can see on the list there. Okay, so you can see that list shows the numbers going up. Okay. First question. I've got Natalie as a first question, a per person on my list. Let's say you have an investment of money and it's doubling every, what do I say here? Every 6 years. How long is it going to be before you're a millionaire if you invest $100?
Probably at least 6 years.
We're over here for a while. Yeah. I was gonna say. I think in that range.
You come talk.
84 years? 84 years. How'd you get that?
Take a million divided by 100.
You said that.
So doubling every 6 years, how many doubling periods did you get?
You have to get it.
51,000.
14 doubling periods? Okay, thanks. So in 14 times 6, yeah, that's 84, right? So in 84 periods of doubling time, you become a millionaire if you 84 years. Put in $100, 84 years from now, you get a million. So question 1, 14 doubling periods.
Freedom 84.
I'm on Freedom 96. Okay. Question number 2. Very similar. Let's say your bacteria doubles every 20 minutes. Now you've got 1,000 CFUs, 1,000 colony forming units. What's
9,
next?
10, 10.
10, 24. How many doubling periods before? Oh, yeah. Jen.
See, I'm confused now because I had it as 180 minutes with 20 doubling periods.
How many doubling periods did you have?
And so I might have done my math wrong, but I have it as 180 minutes to get to a million.
180 minutes.
Oh, it's 20 minutes.
And as we say, how many doubling units did we have?
Times 20.
So you're saying there's 9 doubling periods? 1, 2, 3, 4, 5, 6, 7, 8. Is that 9?
No, that's 10.
So that's 3 hours.
9 is to the 512.
Yeah, as we see, you have to find 12 down the bottom. and
9.
9.
So you might have missed one.
That's correct.
Okay, it's just a.
No, damn well.
Yeah, so looks like 10 doubling periods for this question. Thanks. Adam would be the next person I would ask, but he's not there. Danielle, your question. Okay, we have B. cereus, Bacillus cereus. This is the gravy bacteria. The one that if you have gravy, no, wait, rice. This is the rice bacteria. Perfringes was the gravy bacteria. Bacillus cereus is the rice bacteria. If you have that on the counter too long, it can double and it can make you sick. And it has a doubling time of, let's say, 30 minutes. Okay, let's say you start with 200. That's okay. How long before you have 10 to the 5 bacteria? 1, 2, 3, 4. 1, 2, 3, 4, 5. So 200, and it doubles. This is 4, 4 here. And this is the stopping point right there. What's that?
Possibly 217 minutes.
1, 2, 3, 4, 5, 6 doubling period, 6 times 30 minutes each.
I didn't get the question. You said what the question?
What is the question?
Yeah.
If you have 200 colony forming units and it doubles every 30 minutes,
Okay,
how long before it ends up being 1 times 10 to the 5? So
that is 100,000.
100,000. 1, 2, 3. Oh, this is,
1,800.
100,000.
Oh.
So that will be.
so I didn't quite get there yet. One more.
There should be 9 doubling
1,
periods.
2, 8, 9 doubling periods.
which were
And 9 times 30 minutes,
270
270. Thank you. Well done. Okay, you got it figured out. Question 3, question 4. Tobechku. You start with 10 to the 4 bacteria per mil. 10 to the 4. How long is it going to take to make 10 to the 8? So let's just, yep. 10 to the 4 is 1, 2, 3, 4, 4 zeros. So it doubles 20,000.
Okay, there has got to be an easier way to do this.
This thing has an equation, the W equation P. Let's use the equation. Let's use the equation so that we don't have to be.
Oh, I put the answer up there.
I was gonna say, you do realize the answer's up there.
If this is a Chris question, it would be Martian days.
I'm trying to say, let's take out the guesswork out of it. And use the mathematical method to do it. Let's use the exponential equation. So yeah, do it.
I was gonna say, I know there's an equation to make our lives a lot easier than writing all these out.
Yeah.
I don't remember what it is right now.
Isn't AE to the,
Is P. The
RT and something like this? It's something like this. And I know
Yes. 1 plus
there's an
R
equation
divided
for that, but
1 plus R.
So as long as you're expecting us to essentially go through and write it out, you'll give us time to do that.
I wouldn't even ask this on a
Okay.
test. This is just kind of like,
I'm going, this is
us
gonna
having
take
fun.
me too damn long.
yeah,
So here's your formula if you want it.
okay.
it looks something like what it will be is when we use the. Where
Here. At the bottom.
this one, because this is one, so what's the value? 210. What it will be is
Oh, yeah.
it original amount.
I did it in class.
This will be the equation now.
I could have done it before.
I think so.
Yeah, it should be something like that.
That's right.
I'll leave that to you if you want to try. I'm happy with your trying it out. Okay. Thanks.
Get a free coffee tomorrow.
Okay. That's my little introduction explanation about exponential growth. So the idea is that it is exponential. This is what was really scary with the pandemic, is because at the beginning, remember 2 years ago, numbers of people getting sick started becoming exponential. So it wasn't just like day one, it's 100, day two, it's 200. It was more like in 3 days or 4 days, it doubled. So that's why it'll really scared people. Okay, so the next section, I'm talking about temperature with microbes. So there's a couple of words that go with temperature for being an environmental factor for growth. You've got the optimum temperature. This is like the temperature that your microbes like the most. Then there's the minimum, there's a maximum temperature, and there's 3 words that go with what kind of temperature these organisms really enjoy. There's the cold loving bacteria. So it's based on a Greek word, psychro, meaning cold, and philic, meaning it likes it. So there's psychrophilic. These bacteria like to grow in the cold. If you know of a microbe that likes to grow in the fridge, that's your psychrophilic organism. You think, what grows in the fridge? Really, nothing grows in the fridge, right? No, stuff grows in the fridge all the time for me. I get mold. Mold grows in the fridge. So all kinds of fungi grow in the fridge. And there's microbes that like to, that live in the Arctic as well. They live in cold lakes, they live in deep waters. So there are psychrophilic organisms. Next, middle, what we consider to be the middle. It's like room temperature to about 30 degrees. Middle living, 30 degrees to 37, it says there, but I think I should have put down maybe 20. Anyway, so there's a middle ground. These are referred to as mesophilic, and then there's thermophilic. These ones like hot environments. So meso, thermo, and psychro. And if you take a look at the temperature ranges, you have psychrophiles, they like the cold temperatures, mesophiles like medium temperatures, thermophiles like warmer temperatures, and then there's your extreme thermophiles, something that you'd find in some place like the national parks in Wyoming, Yellowstone.
So they grow more.
Yeah, and they can grow up to in waters that are over 100 degrees Celsius. So there's extreme.
More. Generations per hour, more per hour.
I don't know what this line is actually representing on the left.
I was gonna say we,
Generations per hour. Yeah, you're right. Okay, yeah. So it has an exponential phase.
which would kind of make sense, because those extremes you don't stay in very long normally.
Normally.
The faster growth,
Yeah.
and you want to get there as fast as you possibly could.
Oh, they go faster at higher temperatures, slower at slower temperatures. So they can, I have here bacteria grow at a variety of temperatures, but it's also fungus as well. And I'm just repeating stuff here. There's organisms that grow close to freezing temperatures, meso temperatures, and thermophiles as well. Psychrophiles, isolated from the bottom of a cold lake, maybe anywhere from 0 degrees to 4 degrees. And the question is, why should we be worried about psychrophiles? Natalie's got an answer. And so does Paul.
Because
What's that?
Maybe with global warming, there's more freezes and frozen dormant for a long time.
because
Maybe
the refrigeration will not, refrigeration will not affect them. So if you try to stop them, they will still be growing.
And this is the problem with Maple Leaf Foods, is they had organisms that didn't stop at 4 degrees Celsius. They still grew.
I
And
was
listen-
gonna say,
Yeah.
Food safety.
Oh, and I have here what food is, what bacteria is a concern. So what was the bacteria from Maple Leaf Foods? You guys, yeah. Mesophiles.
It's like the-
They're the most common bacteria. There's saprophytes, which grow on dead organisms, parasitic microbes, and they grow over a wide range. Pathogenic species, human pathogens typically grow at 37 degrees. And there's a wide range of them. Saprophytes have a wider range than parasitics. That's interesting. Anyway, 2 species have an optimum at 37 degrees, but one could grow better between 5 and 45, and the other is growing between. So it's dependent upon the organism. You could call them a mesophile, but their actual preference might be different than another mesophile. And why do we concern ourselves about mesophiles? Is because we're mesophiles essentially, right? Okay. Last, keep going. Thermophiles, they're found in hot springs, heating piles, manure. Anybody ever, do you guys work with manure? Yeah.
Do we?
Okay, so yeah, you've had experience with compost and manure, and you know that it doesn't just exist at room temperature. This stuff gets hot. Like it gets,
Same with like silage.
Yep.
yeah, ferments especially.
And why you have to be so careful putting like wet hay and stuff in a building. It
Explosive.
becomes really bad.
Granary fires, barn fires, yeah, silo fire, yeah. Problem is in pasteurizing milk because they grow at the temperature that they're intended to kill. We have Streptococcus thermophilus. This organism doesn't die at the 70 degrees. It can still be there in part, but it's a beneficial microbe, so we don't worry about it too much. Or I think it might be 50 degrees. Anyway, some cause canned foods to spoil because they're forming thermoresistant spores, and the organism of real concern that we always worry about in high temperature is
Botulism?
botulism, Clostridium botulinum. Paul, you were talking about tetanus the other day, lockjaw. And I was wondering, like, for some reason, it was kind of triggering a memory for me, but I remembered that it's Clostridium tetani.
Yeah.
And both Clostridium botulinum and Clostridium tetani, they both have the Clostridium part of the name. So
We're the same.
same great big family, yeah.
Similar outcome.
Okay, here's some temperature questions.
And the same.
Okay, everybody on my list except Adam and Lindsay have answered, but I leave this question open to everybody else. Which mesophilic bacteria was studied by Robert Koch? Causes consumption. Yeah, go ahead, Jen. Right. What's the name of the eukaryotic microbe that's capable of fermenting juice in a psychrophilic environment?
What's it mean?
Eukaryotic ferments fruit juice. Yeah. Yeah, Danielle?
So how is that psychophilic?
It's cooler.
So what
So-
constitutes cooler? Because I would have thought that would be more of like a middle range temperature.
5 to 15. You want your wine to be at a cooler, like a refrigerated almost temperature for slow fermentation.
Okay.
So it's arguable that maybe it's not a perfect psychrophile. Yeah. Because we use it at room temperature to make bread too, right?
Right, that's what I was thinking of it as more room temperature, but I guess, okay. So
Yeah.
it's one of those that could be both.
It has a range.
Yeah.
Yeah.
What's the low end of that range then?
For psychrophiles, it's like down to 0, proper psychrophiles, but for the sake of the question, I was thinking maybe 10, 15
Is
degrees.
there a
12.
category below 0?
Can anything survive below 0?
Yeah, I think those are the quick
Of
ones.
course.
Can anything replicate itself? below 0?
These organisms are really weird. They have, antifreeze within their cells to propagate and maintain themselves.
Is that category?
I don't know.
Is it
not sure.
Thermophiles.
Pardon?
Is it not extremophiles?
It could be like a type of extremophile, yeah. Not sure what it would be called though. Like it must have a name. It must be in a category. Two simple methods for preventing food spoilage, temperature responsive microbes. What are two of them? Refrigeration, pasteurization. Yeah, good. Okay, so that's the temperature. The next part of the presentation is the oxygen. With oxygen, I talk about 2 words for having oxygen and no oxygen. I talk about the different kinds of aerobes. Let's take a look at that. Okay, so there are what are referred to as environmental factor being the oxygen effect. If it's an obligate aerobe meaning it likes oxygen, obligate meaning it's obligated to be living in an environment with oxygen. Obligate aerobes need oxygen. So you'll find them at the top of your media. There's a special media that we get people to use called thioglycolate. What you do is you take a streak of microbes, stab it into your thioglycolate media, and then let it sit for 3 days to a week maybe, and then you take a look at it again and find out where the microbes are living. If they're on top, they're the obligate aerobes. If they're obligate anaerobes meaning they don't need oxygen to survive, but they survive well. Obligate meaning they're at the bottom. Okay, so obligate top, obligate bottom. Aerobe on top, anaerobe on bottom. Then there's what are referred to as facultative anaerobes. Facultative meaning it can live with or without oxygen. So something like Escherichia coli is a facultative anaerobe. If you put it in thioglycolate media, it'll be on throughout. You'll see bacteria on top, bottom, middle, everywhere. And then there's also aerotolerant anaerobes. Now this is another term meaning very similar to the same sort of thing. It tolerates air. And then there's a 5th category, micro aerophiles. Micro meaning it stands only a little bit of air. So it's not obligated for oxygen, but it needs a little bit, but not too much. So that's why you find them just below the surface of your media. Now, can you tell me the name of a food that we studied in lab where you saw obligate aerobes growing? Yeah.
This movie's just under the surface.
Yeah, kombuchas. You remember the kombuchas right on top? It floated there. So that would be your obligate aerobe. Yogurt? I think that would be a facultative because it was throughout the milk. Yeast. Does it need oxygen? I'm trying to think. we stopper the carboys. Oh, it's emitting gas, but
In general, we're not supposed to lock it, 'cause yeast can survive in the presence or absence of oxygen.
so it's facultative. But I don't know if they describe yeast as facultative because it's
Some
a
yeast are
fungus.
Some yeast are top fermenting, some are bottom. So for the ones that are top fermenting yeast, it should be
Obligate, probably.
obligate. For the ones that are bottom fermenting, it should be obligate anaerobes.
Right, so they can be kind of both. Okay. And this slide essentially repeats everything. Aerobic organisms survive in oxygenated environments. Obligate aerobes need oxygen to grow. They use oxygen to metabolize everything. Facultative anaerobes, they use oxygen if it's available, but they have anaerobic methods of making energy. Microaerophiles require some oxygen. And aerotolerant don't use oxygen, but they're not harmed by it. Next slide here, oxygen effects. An anaerobic organism or anaerobe is an organism that does not require oxygen for growth. It reacts negatively or can die if it's present. And it says here, an anaerobic organism can be unicellular. Unicellular. It could be a protozoan. It could be one of these things that live in water that are totally eukaryotic, or it could be a bacteria, or it could be a worm, or some sort of parasite. Oxygen effects. Did you have a question?
I'm sorry, I meant to finish.
We can-
It was about yeast.
Yeast, yeah.
It's both, because it's both aerobic and anaerobic respiration.
It depends on what you're using it for and which one you're using.
Right.
Yeah, and also just what you do to it.
And-
Restricted oxygen, it will just start.
And likely strain dependent too, because there's so many different kinds of yeasts out there. Treat them gently, especially if you need it, right? If they say use it in one way, just follow directions. There are three categories for oxygen effects: obligate aerobes. Sorry, obligate anaerobes. They're harmed if you expose them to oxygen. Aerotolerant organisms, they can't use oxygen but tolerate it. And facultative, they can grow without oxygen, but if you present it there, they'll survive there. Here's a picture. As a class, go ahead and match. You can't see my list of people's in the way. Can you identify the obligate aerobe?
Fourth line from the right.
So
Fourth if you count it from the left.
A
Second if you
is
count it from the right.
fourth from right.
Left, fourth from the left, or second from the
Let's just do
Oh,
from
sorry,
left because
from
we
the
did
left.
right.
From the left. A is the fourth one. Obligate anaerobe. Which one's?
Two.
Yeah, I'd agree with the Dutch group. The third one. You can see the plug is at the bottom. So B is third. Facultative anaerobe. Throughout. Can you tell?
The last one, five.
I'd agree. The last one is as well. Could also be the first one. Maybe.
Yeah.
Definitely the last one. For sure, I agree. The facultative anaerobe's the last one. The first one with the white material in it, it's hard to say. It's more. C is definitely, C, facultative anaerobe, definitely the fifth one, but I don't know about the first one. It's kind of hard to say. And this is the thing with the lab. You have these definitions and you have these real things. Can you match the definition with that? Sometimes it's kind of dicey. And I think that's why I wanted to show you the picture. Is kind of, you kind of have to make a judgment on it. Aerotolerant anaerobe D. Aerotolerant. It could be the first one, maybe. I think obligate aerobe is a second to last one. That's the fourth one.
Second.
Oh, the second one, you'll see has a very, very thin line just below the surface. So that's the micro, that's E. Is that what you're saying?
I thought, yeah, since it was just below the surface, so where do you see that line?
Yeah. So E, that'd be the second. Everybody agree? Okay. Okay. So that's the idea of oxygen and the words you need to kind of describe oxygen use in microbes. Then there's pH. With pH, we have organisms that live at high pH, low pH, and neutral pH. So if it's low pH, like less than 7, that's an acidic organism. If it's higher than 7, then it's alkaline. Oops. And if it enjoys living in an acidic environment, we call it an acidophile. So if it's low pH, acidophile, high pH, alkalophile. Neutrophiles, they live on meat, plants, kitchen utensils, fruit prep areas. That's a very common microbe. Alkalophiles, things that thrive in an alkaline environment where the pH is very high. I looked it up. There is one lake in Saskatchewan called Manitou Lake or Little Manitou Lake as well. They're resort places, probably one of Canada's few salt lakes. They have a very alkali environment. Have you ever been there? Anyone? No? I have this vague memory as a 4 or 5-year-old going to one of these pools, and people would go there as a healing sort of area. They'd go for their sore bones because it was also a warm pool as well. And then there's acidophiles, and they live in acidic environments. And you think anything that can survive vinegar, that'd be an acidophile. Anything that generates these gluconoacetic bacter type bacteria or Acetobacter aceti, the one responsible for most of vinegar production. Yep. Anything that makes your apple cider vinegar. You can collect halophiles from vinegar, or sorry, from pickles. This is something that people sometimes do. Yeah, question? Less than 7, sorry. Acidic conditions. Sorry, pH, I say pH 3 or below, but it's below 7. That's going to be your acidic bacteria. So 5, 4, 3, 2. One bacteria or a group of bacteria that are really harmful are the bacteria on your teeth. And what happens when you eat is bacteria become more active. Because at the moment, you don't have any bacteria in your mouth because you're not eating, you're not drinking. And then as soon as you eat something that's sweet, bacteria, they become active and they start generating, they're metabolizing the food. And when they metabolize the food, they're also decaying your enamel at the same time. And so this is how cavities form, is you have children who, or adults who eat, bacteria are active, period of time goes by, you eat again, bacteria are active again. And this is referred to as a Stevens curve, where when they're active, they're generating acid, and then they become inactive. You eat, they become active, and you can actually follow this. This sort of effect was known in the 1920s. This is how long ago it was actually observed. 1920s, 30s, where the pH in your mouth actually drops after every time you eat. Okay, so, and I had a question, ask another student about pH and microbes. Or explain a problem encountered by frequent snackers. Let's say you snack all the time. You're always chewing something that's food-like. What could possibly happen? Question, yeah? Diseases?
Question.
Well, it depends on what you're snacking, because if you're always eating candy, it's really hard on your teeth, but it's also sugars that can be hard. It can be hard if you're on the verge of becoming diabetic. And if you think about it, the people who lived hundreds of years ago, they didn't really have an idea of brushing. They didn't have an idea of maintaining health. So they probably survived with all kinds of.
So like gingivitis and stuff like that.
People have been brushing their teeth since like Roman.
The brush, they use like there's a tree in my place, there's a tree, you cut the branches, there's substance and use to brush. It's antibacterial.
And a lot of wood
The
products
tree-
are, yeah.
Yes, the wood is antibacterial when it eats. That's what they use to brush, not the kind of brush.
And it's effective.
It's effective.
Yeah. People have good teeth. Yeah.
It's better than you think.
Yeah. Because it's antibacterial and then it also cleans. It
My mother-in-law drinks coffee with 2 or 3 teaspoons of sugar, maybe 4 teaspoons of sugar. She's in her 80s, but she's like a hummingbird. She's, yeah, I think so. Coffee and sugar, she's always.
My great grandmother, she had like boxes, and the fridge was just chocolate. Chocolates, and then the freezer was always. She lived till like 90.
So, yeah, good or bad, this depends on the person, I think. Diet. Okay, and the last thing that I said I was going to talk to you guys about was this water activity measure of bioactive water, bioavailable water. It's the ability, it determines the ability of your microorganisms, can they grow? And so it's this number, and expresses a ratio of vapor pressure in a food to the vapor pressure of pure water. So P over P naught. So it tells you if the water is going to move into the food or out of the food. It predicts whether it is likely to move from the food product into the cells of the microorganisms that might be present. Drying or adding salt or sugar can change your active trait. Okay, so you guys are familiar with that. So here's a picture of water activity. So if your water activity value is 0.6 or lower, you will not have any growth. Pardon?
Honey is on that side as well.
Really?
Yeah.
It's because it's so much sugar. Ah, okay, makes sense.
But I don't think that's what honey uses. Honey, I don't know, because I've read that the bacteria gets inside the honey, it will draw the water. It will draw the water from the bacteria, and you have lost the cell. It draws water out of that bacteria because of the high sugar.
It depends on what kind of cells that are in the honey, I guess, because remember, Clostridium spores can survive in honey. And that's why we don't feed the honey to babies.
Yeah, that's one.
But regular bacteria, yeah, you wouldn't be able to keep regular bacteria in honey.
I don't know whether it's water activity, but I think it is not because of the high sugar. Because honey is that we measure here contains 17% water.
But there's the availability was very low.
Yeah.
I was gonna say the water availability is very low because it's tied up with all the sugar. Maple syrup's the same thing. It falls above or below the 6 as well.
Oh, yeah?
Because it's
Cheese would
so
be the
much.
opposite of that. Cheese has got a low water content, but a high
Right.
availability.
Yes,
Oh,
because
okay.
it's not bound anywhere.
And I have here milk beverages around 0.9, evaporated milk, tomato paste, fairly high, and then down to less than 0.9. So you get your gram positives at around 0.9. Other cocci are between 0.85 to 1. And then mold and yeast can survive everywhere. Oh, 0.6, yeah. So you guys have heard of these things, these numbers before? You're familiar with these? Okay, so thanks. Helps me to kind of go over it. So here's some more foods. Cabbage is about 0.6, carrots about 0.6, horseradish 0.7, potatoes 0.6 to 0.7, strawberries 0.6 to 0.7. So they have a water activity number associated with them. So if you add sugar to help them, you'll preserve them. So this is why we have jams, is you just add sugar to strawberries and you can preserve them for a long, long time. Acidifying it can also help. So if you add acid to your cabbage, like you would you? Pickled cabbage, yeah. Oh, pickled carrots, pickled cabbage, pickled horseradish,
Pickles.
and pickles. Speaking of pickles. For you guys, anybody who's wants to try my pickles, you go here. Well, it's not a competition, no, because I don't know if they're what you think. All dill.
We had this conversation now.
So we'll try some. It's
the big law.
food spoilage prevention.
In between hamburger and Bunker, right, in Frankfurt.
Food spoilage, what am I saying? There's a strategy inhibits spoilage by citric acid, benzoic acid, tartric acid, sugar, fructose. Oh, there's a lot of different ways, even spices, vanilla, garlic, oregano can help preserve stuff. And here's a big chart for limiting growth and what kind of organism is limited by the growth. So if you want to take a look at that later at your own time, there's not going to be a test question on the final test about water activity because I just went over it real quick, but you guys have any questions? Okay, I'm going to go over the assignment with you guys. And sadly, Aaron, Hamish, I don't have a video for this for you guys, but
Take a picture after.
yeah, that's a good idea. So I'll say goodbye to you guys now. Unless you guys have any other questions. Aaron says no questions, thanks. And Hamish, okay, so I'll save the presentation and see you tomorrow. Okay, have a good day, guys. Bye. So I'll end the